Dear students, our today's topic is anatomy of dicot and monocot stem. Anatomy of dicot and monocot stem. We know in anatomy we are talking of internal structure. Then before this we have discussed anatomy of root also, and today we are going to discuss anatomy of dicot and monocot stem. First of all, when we draw the structure of dicot stem, at first we see structure of dicot stem. We will try to see what is the arrangement of vascular bundles and what about their shape. Arrangement of vascular bundles and what about their shape and size. For example, this is a vascular bundle and these are arranged in a ring. Vascular bundles are arranged in a ring and in between there is pith. This is pith. And first of all, we will see that vascular bundles are arranged in a ring. Vascular bundles are arranged in a ring and in between there is cambium. In between there is cambium. So this cambium is called as vascular cambium. It is called vascular cambium because it is present between xylem and phloem. Xylem and it is xylem and it is phloem. Then we know that these xylem is to when xylem is towards pith, xylem is present. When xylem is present towards pith and phloem is present towards periphery. So this arrangement we can say it is conjoint type of arrangement. So we can say conjoint type of arrangement. So we can say there is conjoint type of arrangement because xylem is present towards pith and phloem is present towards periphery, and we know that cambium is present between xylem and phloem. So this is called vascular cambium because it is present between xylem and phloem, or it is present between vascular tissues. This cambium allows secondary growth. This cambium allows secondary growth, and we know that these vascular bundles are arranged in a ring. Vascular bundles are arranged in a ring. Now what about shape and size of vascular bundles. We know that in case of dicot stem, shape and size of vascular bundles is same. Shape and size of vascular bundles is same. Now we will draw the structure here. For example, we know that there is also outermost layer which we call as epidermis, and in every structure I had told you this epidermis is outermost layer. Epidermis, it is outermost layer, and I have told you it is single layered. Outermost layer, and it is single layered. Now there are some extensions, or we can say some type of outgrowths, which we call as trichomes. These trichomes are multicellular and these are multicellular, and the function of trichomes is to prevent loss of water. Their function is to prevent loss of water. Now we know that below the epidermis are parenchyma cells. Below the epidermis, these are parenchyma cells. These are parenchyma cells. So these are parenchyma cells, and the region of this is called as cortex region. Cortex region. Below the epidermis or these are collenchyma cells. These are not parenchyma cells, these are collenchyma cells. Collenchyma cells. We know that below the epidermis there are collenchyma cells, and after that we have parenchyma cells. For example, these are parenchyma cells. Parenchyma cells. And then there is the layer of endodermis. Then there is the layer of endodermis. And we know that this endodermis is situated in also cortex region. So we can say it is called endodermis. We know that this cortex region is divided into three parts. First is collenchyma, second is parenchyma, and the last one is endodermis. Now we know that I forgot to tell you that in case of vascular bundles, there is array which derives from one vascular bundle from cambium to another. It is called as medullary ray. Medullary ray. So we were here we were talking of endodermis. This endodermis, we know that this endodermis contains storage. Storage. So it is called also storage sheath. It is called storage sheath. And there is a structure which we call as pericycle. Pericycle. What is this pericycle? This is the region which is semilunar in shape. The region which is semilunar in shape. Semilunar in shape, or we can say moon shaped. That is moon shaped. Then there is also region which is called as semilunar, which is also called pericycle, and this is semilunar in shape. For example, it is moon shaped. Then there are cells which surround this. There are some parenchyma cells present, for example, which surround this. Parenchyma cells. These are parenchyma cells. Then there are vascular bundles. As I have told you, these vascular bundles where pith is present in between them, and there is vascular cambium, and it is xylem and phloem. So it was the structure of this dicot stem. Now we know that these vascular bundles are of open type. I have already told you these vascular bundles have open type of vascular bundles. I have already told you that there is conjoined type of vascular bundles. So we know that a ray derives from vascular cambium, which is a parenchymatous ray. Vascular cambium, which is a parenchymatous ray, which is parenchymatous ray, and this is called a medullary ray. And this ray allows the lateral growth, which result is the formation of a ring or appearance of a ring. So vascular bundles are arranged in a ring. Now we know that I have told you these extensions which come out on the surface of on the outermost on the surface of epidermis. These are called as trichomes. And we know that below the epidermis are thick walled cells, which we call as columnar cells. Sorry, I have told you parenchyma cells, but these are columnar cells. And I will repeat it again. Below the epidermis are thick walled columnar cells. And we know that the region below the epidermis is known as cortex region, which I have told you, and that region is composed of 3 different types of cells. So cortex region is divided into 3 different parts. In case of or different layers in case of stem. Collencama layer is called hypodermis. Collencama layer is called hypodermis, and then parenchyma layer is middle layer. It is called cortical layer. It is middle layer. It is called cortical layer. Then third layer is cortex region, which is called as endodermis. Just closed endodermis or certain specialized structures which can be of sclerenchyma. So they have a way of semilunar arrangement, which I have told you, having moon shape. This region is called a pericycle. Now endodermis cells contain storage, so they are called storage sheath. Then there are vascular bundles, which are in the form of ring and which are having conjoint arrangement. And vascular bundles are of same shape and size. These are also of open type. Now we can talk in case of monocot stem. Monocot stem. It is monocot stem. In case of monocot stem, I will draw here the vascular bundle. For example, these are vascular bundles. And some are small and some are large. So we can write that this, in case of monocot stem, size of vascular bundles vary. So there is vascular bundles are of different shape and size, and we know that in center there is pith. In center there is pith. So we know that these are of closed type vascular bundles because they are not having cambium. So we can write here no in cambium in case of monocot stem. So at center vascular bundles are larger. So we can write here vascular bundles are larger at center. Number 2, vascular bundles are smaller at periphery. Then there is arrangement of vascular, so we can say arrangement of vascular bundles is irregular. So there is an irregular arrangement of vascular bundles. So arrangement of vascular bundles is irregular. Then we know that there is no cambium, so they are a closed type vascular bundles. So we can write here they are having no cambium, so they are closed type vascular bundles. Now when there is no cambium, when they are closed type vascular bundles, so automatically we can say there is no secondary growth because of absence of cambium. They are having no secondary growth. No secondary growth because of absence of cambium. Then we know that there is also conjoint arrangement of vascular bundles. There is also conjoint arrangement of vascular bundles. Now we know that these, in case of trichomes, there, or in case of monocot or dicot stem, the number of trichomes was large. But in case of monocot stem, there are less number of trichomes. In case of monocot stem, they are all having less number of trichomes. And the function of trichomes I have already told you. It is, then there is size of vascular bundles. Size of vascular, so we can say size of vascular bundles can vary. Size of vascular bundles varies in case of monocot stem. In case of monocot stem, then we know that vascular bundles are present at pith. Vascular bundles present at pith are larger. Vascular bundles present at pith or larger, while as vascular bundles present at periphery or smaller. So it was all about your anatomy of dicot and monocot stem. Thank you very much.